Okay, we'll go ahead and get started. We're good? Yeah, so thanks everyone for coming to another data integration meeting. The goal for this session is to start to think through the different ways people are trying to do data integration and different avenues or perspectives they're taking to try to support or refute predictions. Because a lot of the reason we're doing data integration is to have multiple lines of evidence to either say whether what we think is happening in biology is actually happening or not. So this is the lineup of the talks today. I wasn't quite sure of the scheduling, so we will have a break hopefully for about 10 minutes so everybody can refresh, get more coffee, get some more snacks, and then we're hoping to finish by 5:30. Okay, so I'm going to do a quick introduction. Why bother with data integration and why does your data matter? So if you went to the data integration talks, I think Leanne and Kirsten and Adam did a really great job of starting to set up why we should bother with data integration. What is that going to actually enable? And so this is actually a slide from Adam's talk as well that I'll go through again in case you missed it. The FAIR data principles is a start, right? So making data findable, accessible, interoperable, and reusable. But there's nothing in there that's specific to a particular domain, and that was done intentionally. So after you find your data and you manage to get it all into some location where you can do something cool with it, you still need to unpack those files, figure out what's in each one, and understand how they relate to each other. And that data integration part, that data cleanup part, still is a lot of work. And so what we're trying to do now is actually go beyond FAIR and build out this concept of coping with big, complex biological data. So COPE is, again, comparable, organized, predictive, and engaged, where we can actually open up those data files and understand if they can be compared or not. Are they of the same values? Do they have different paths to get to the same answer? For example, you might annotate taxonomy two separate ways. Do they agree, and what do you do if they don't agree? Organizing and creating shareable data, actually putting contextual links together. And so you can see on the right, we have a very basic schematic of a series of observations. Maybe you have chemistry from the environment. Maybe you have organisms that you isolated from the environment. And how do you actually link those together, link that data to measurements in a way that has biological context, so that you can then start to predict new knowledge. So once you actually are able to navigate and query that relationship, you can actually start to say, what do we know, what do we not know, and start to understand if you can propagate across different systems that Adams was talking about. I'm just going to keep talking. And then finally, the goal is to get to a place where we can engage the community. Help us understand where these models fall apart, help us understand where there's gaps in those models, figure out a mechanism for testing and validating those. And so a lot of the talks that we're going to have people walk through today are actually going to be related to that. Now the green button's not going. There we go. A lot of the talks that you're going to hear next are going to be related to different ways that people are actually starting to test some of those models, test some of those assumptions in those models from different perspectives of aggregating and integrating data. So with that, I'll pass it off to Dehal.
Thanks, everyone. Thanks for coming to the bitter end here of the day 2 of the long week.
Talk into the microphone.
Oh, sorry. Everybody hear me now? Yeah. Thanks for the chance to talk about some of our work on sort of integrating computational approaches with phenotyping, experimental phenotyping approaches to improve genome-based prediction. And this is a collaboration that was started between PMI and Enigma and KBase here. And a lot of people contributed to the sort of the ideas and the approaches that I'll talk about, and they're listed here. So I just wanted to make sure to acknowledge those work of everyone here before I start talking about this. And so as I said, this was a collaboration, really got started between Enigma and PMI. And Adam sort of already talked about this in the earlier talk about how these are two very different environments that we study. In PMI, we're interested in understanding the rhizosphere microbiome and how plants and microbes interact to carry out functions in the rhizosphere, particularly with respect to poplar trees, is our system of study. While Enigma, which is really interested in the subsurface microbiome, a very different environment, but we both have common goals of really trying to get to a predictive understanding of these systems that will enable us to engineer these systems and manipulate them for our own use. And both projects have taken sort of over the years top-down approaches, classic omics approaches to understand who's there and what functions they might be doing. But another thing we have in common is that both of us has also taken reductionist approaches, and that includes isolating strains from these environments, generating genomes, studying their physiology in the lab. And so this has enabled us to be able to do phenotyping in the lab of these strains. And so here in PMI, we have a culture collection. And thanks to interaction with JGI, we've generated genome sequences of many of these strains. And what we really want to be able to do is start to use these organisms and their genomes to predict things like community assembly, niche preference, or metabolic resource overlap between these strains. But it's still challenging to be able to do functional predictions from genomes directly. And so things that we can measure in the lab and we have measured is inhibition phenotype. And shown on that plate on the left side there of the slide is some screens we've been doing to look at inhibition across our culture collection to sort of start to get at what role inhibition or antimicrobials might play in community assembly and syncoms in the lab. And so we've run thousands of these pairwise interactions now. And if you heard John Lagergen's talk on Monday at the very end of the session, John is helping us sort of generate AI tools to be able to automate the analysis of these images quicker than we can do it by hand. And so we've just started doing that with John. He's already made some good progress. So that's been great. We've also been looking at constructing syncoms in the library, in the laboratory, and selecting those and looking at metabolic interactions in the lab. And so we'd also like to look at growth and carbon utilization of these strains. And that's a phenotype we can easily measure in the laboratory. But predicting these, as I said, predicting carbon utilization based solely on a genome sequence is still challenging, even today, even with all the data we've generated over the years. And so in collaboration with KBase and Enigma, we've come up with this process to be able to do a combined experimental phenotype and machine learning approach to classify carbon utilization so that we can to develop a classifier and use that to predict carbon utilization across here. And so one of the challenges with this really is incomplete or inaccurate genome annotation, which makes model predictions inaccurate, and also the lack of sort of large standardized training data sets to be able to get this data. And then after listening to the session before this on finding data, I will say it's not easy to go out and find. I know there is a ton of carbon utilization phenotype data out there, but it's not trivial to find it and actually link it to organisms that we know have high quality genomes on that. So even that was just a challenge to do this. And so this is the process we had for this, and this is really, as I said, experimental phenotypes that we've been generating in PMI linked with KBase to help get this process running and the machine learning approaches to do this. But before I go into the details of this pipeline, I also wanted to sort of take a little side vignette into developing standardized phenotyping training sets. And this is where we really got interactions going with Enigma. And we've talked about, okay, what do we need to do to look at reproducible growth phenotypes across institutions? It's one thing to reproduce your own stuff in the lab, but how common is that? And this really got started with Adam calling and asking for a strain for my collection, and that when he was looking at some of their metagenome data and mags they have from their site, actually there's a very prevalent mag in their system that's a Rhodonobacter strain that they've never been able to isolate a strain that matches that mag. And there's actually a strain from our isolate collection from Populus rhizosphere that was a really good match to their mag. And so he asked for it, and then it got us talking about sharing our resources and developing this standardized phenotyping platform. So what's shown here on the right is a phylogenetic tree made by Chris Neely here of a combined isolates from PMI and Enigma that we have high quality genomes for. So it's a tree. Taxonomy was based on GDDB analysis. And then what's shown around, I don't know if I have a pointer here, the outside ring with the red and the green are the strains that are from, so in green are the strains from the PMI culture collection, and red are the strains from the Enigma collection. And then we did some functional analysis with running DRAM and KBase on these, and that's sort of shown in the outer rings of this, just for nitrogen, sulfur, and some Kzyme functionals. And then for those strains that we actually have existing phenotype data, those are shown in the outer ring in blue. And as you can see, we don't have great coverage all the way across the tree. So that was one thing, was to sort of ask the question, okay, can we take strains, swap them between our labs, and reproduce results in base one? So how do we pick the strains? And so we sort of picked these based on phylogenetic functional diversity and also prevalence in our field data. As I said, whether or not there are ASVs or MAGs that match some of our isolates, that we have high quality genomes, they possess traits that are of interest to either of our projects, and then I put well behaved. So we, within PMI, have a large actinobacteria collection too, which includes streptomyces, and a lot of those are filamentous organisms that are not easy to measure growth rates on in here. So for the purpose of this, we stayed away from those and tried to look mostly at just planktonic cells that we knew would be well behaved. So we picked 12 strains for this initial test, 6 from PMI, 6 from Enigma. We swapped those strains, so both locations had all 12 strains. We decided on 2 standardized media, R2A media, rich media that we were pretty sure all the strains would grow in, and then a defined minimal media developed by the Northern Lab that we could then substitute carbon substrates if we wanted to. We are using high throughput plate reader to do these, the EPIC 2. Both locations have the exact same model of plate reader. We tried to standardize starting ODs, growth temperatures, all the things we could standardize. And then we said, let's just do the growth curves and we'll analyze these for these phenotypes listed here. And so here's the 12 strains. We picked 6 Enigma and 6 PMI strains. And what's shown here on the right is just sort of the qualitative growth phase of these. And as you can see, the curves are not identical here. So we can see one thing is, everything grew that should have grown for the most case, except for this one Beribacteria strain, which was a little bit of a problem child. But everybody else behaved pretty well, but the curves are not that great, which is the point I sort of want to make. There's still a lot of variability between sites in this data. We ran a second trial, basically the same. We moved the strains around to sort of make sure there were no plate effects. The second trial looks better. 2 points I want to make here. One is within lab, the replicates are actually almost identical, which is great. But between labs, there are differences in the curves. And that can be seen a little bit better here, which is basically just a correlation analysis between the 2 labs here, looking at growth rate, death rate, lag, and maximum. And you can see these are not perfect. And so this is just, I guess the point I want to make here is that there's still a lot of variability in these phenotype measurements. And so there's some work to do here to reproduce this. So to get back to our phenotype prediction problem here, and so here we developed this sort of standardized approach to sort of annotate microbes and running them through a machine learning approach with phenotypes that were generated either from the PMI culture collection, where we had phenotypes, and some phenotypes that were collected from the literature, and then a collection of carbon utilization phenotypes from the Arabidopsis leaf microbiome work from Julia Vorholt's group here. And so we had good carbon phenotype data on these strains to use as a training set, and high quality genomes from all of these. These genomes were then annotated with multiple annotation pipelines through KBase that are based either on metabolic orthology, protein functions from UniProt or UniRef databases, and de novo clustering, and then sort of ask which features from these predictions, from these annotations, led to better phenotype predictions, the best. And so here's the pipeline. There's the genomes. There were 626 genomes, I think, that were loaded into KBase. Got 2 minutes left. They were annotated. The features were represented on this and then run through machine learning. And then we asked which features were best at predicting this. And then we also did a combination where all of the annotations were combined together, a combined feature set, and asked which predictions. And so here's a heat map of this. On the bottom is the features, and then on the y-axis is the carbon utilization phenotypes prediction. And the heat map is basically showing at the top of it, in those little red numbers, or which feature performed the best. And you can actually see the combined feature representation by far outperformed the other ones easily. And so with this machine learning approach, we were basically able to develop a classifier for 98 carbon utilization phenotypes. And so now we can go back to our culture collection and our genomes and basically try to validate these predictions by running that classifier across these genome strains and then experimentally verifying that. And that's what we plan to do next here. And also work on improving our reproducibility and throughput of our phenotyping assays. And then lastly, Adam brought this up in his talk too, and I've seen several instances in the media of this now, of creating an accessible resource, or a microbial atlas, I call it, of sort of deeply characterized isolates to facilitate resource and data integration across multiple DOE projects. One thing I realized sitting in most of the talks and looking at the posters, that there are numerous projects now across the DOE portfolio that have isolates with genomes and lots of phenotype data. And I think it's a great opportunity now for us to try to integrate all of those data sets to do something bigger than what any individual project could do with these. And I'd love to hear what others think about that and how that might work in the way we are now. So I'll end there, and I want to thank everyone on the PMI team, the Enigma, and the KBase for the interactions in this project. Thanks DOE for the funding to do this work, and JGI for all the genome sequencing they have done for us. And then I just want to make a plug for Priya Ranjan, who's going to give a talk tomorrow on sort of microbial community modeling efforts. And this was some of the efforts that come out of the KBase PMI SFA funding plus up that we got a couple years ago that really got us thinking about some of this work. And with that, I'll stop there. So thanks everybody for listening.
We have time for one quick question, if there is any.
Hi, Dale. So I guess my question was for the for the carbon sources that you were testing your models against, were those all carbon sources for which the metabolisms are well worked out and the gene lists, might be represented in the annotation, or were some of those things where it might have been less clear as to what the actual pathway was?
Some of those are less clear. A lot of them are well-known metabolisms in those things. But one of the things we tried to do early on was to is to look at that by creating metabolic models for all of those and looking at the RAS or seed annotations to ask how complete was any of the pathways and try to use that to select strains to look at. And also we have selected, both projects now have selected a number of carbon substrates that we would like to start to go after to look at that, the things that we know, we don't know their metabolism, things from our point of view that are in popular root exudates, that might be defense compounds that we are very interested in understanding how they're metabolized.
Hi, my name is Shinjae from the Brookhaven National Lab. This is also another KBase Plus sub project with the PI, Paramvir. Sorry, I suddenly lost the name. So next page. I gotta go back. So the fundamental question of the knowledge extraction is, I'm gonna give an example as the synthetic biology as the science driver for the knowledge extraction. But this is a very universal problem across science domain, across the biological domain too. So we were gonna use two driver cases. One as the synthetic biology, another one is the PPI interaction relationship. But here is primarily focused on the synthetic biology as the main driver. So fundamental question is that how do we accelerate sustainable bioeconomy? That is essentially democratization of the genetic tools and the technology from the energy to the food crop, or like bacteria and so forth. Then the synthetic biology need well-curated genetic tool. The major bottleneck in the synthetic biology is identifying suitable microbial targets for specific application conditions and their genetic tool for cultivation and the engineering of non-model microorganisms. This is inherently blocking our PR mission progress, and then in case of synthetic biology, it hinders the redesign process. However, a lot of the experimental data, the primary outlet of those kind of experimental data is the literature. So we need automated processing pipeline from the literature. And then I think Rick Stevens gave a very wonderful keynote speech on the first day. So I'm not going to go into any of these and explain the concept. I think the natural language processing and then the large language model is the ultimate solution. For that, we want to extract. So we have two technical objectives. First is genetic tool described in the free-form natural language text. And then from there, we want to extract key and relevant information from there. Another primary outlet of the many experimental data is in the form of the table and the figure. So the first one, we mainly use the natural language processing, large language model to extract those information. And then for second one, table and the figure, we are mainly using the computer vision technique to extract those information from the literature. At the moment, this is not currently funded activity, but in the previous cycle, we got funded on doing this and providing the many updates, how much we made the progress. So thanks to the DOE Nuclear Energy Office, they supported the table extraction. For instance, this is National Nuclear Data Center. they manually create literature and extracting table and they put that table into the database so that people can easily search. Over there, initially this work was supported by BR, and then we recently delivered this system to the nuclear energy, and then it is waiting the full production service soon. The way table extraction working is, if you guys see the table here, generally identifying each cell first. Here is like column and the row alignment is very important problem. Then we are extracting each text using the OCR. And then at the moment, accuracy is pretty good, very good. And then once we extract, then usually human validator double checking the automatic extraction tool output. We are using the computer vision based the object detection technique to detect each cell, and then we automated the pipeline we built. Another good news is that when we started working on this project, not much AI-based tool, but now there are many commercial services doing that. So if the BR community are interested in and then don't want to use the commercial service, we have the technology. On the other hand, figure extraction is much more challenging because the figure form is much diverse. Like we demonstrate in the bar chart, and then from the bar chart, another challenge is that very difficult to collect and label exact precise point and so forth. So what we did is that we are using the LaTeX and then synthetically generate a large amount of table figures automatically. And then from there, we trained the AI model. Our technique is similar to the object detection model, but instead of the regular object detection, in the figures, the most important thing is the bar chart. What is the peak bar point value, right? And then all the quantity metric on the y-axis and the x-axis is tracking those points. So we proposed point proposal network. It's in the archive, in the paper, all our recipe. We got several love calls from the startup company. Oh, they want to do the commercialize. I'm not sure whether we will do it or not. So that is another tool we developed. But this tool requires more work, and then it is not ready prime time. So that is update on what we initially started funded by BR, and then moving toward the next step. Information extraction can be done by a large language model to identify named entity and then extract it directly. Or another approach is nowadays prompt engineering, meaning asking questions to the large language model and then make them to answer. Here's the first example is we are doing the protein-protein interaction using the identifying protein named entity, and not only we are not stopping there, we are identifying different protein entity type, right? Different protein entity relationship type, and then detecting them, and then for that we developed some of our unique and novel state-of-the-art method to relationship classification as well. From now on, I'm going to focus on the gears toward the large language model based approach. So this is more geared toward the synthetic biology. If you are seeing the identifying organism host and the genetic tools, the first step is we can just ask the question large language model and then it answer, but we cannot evaluate, right? So the first step is the annotate. The first step is annotate literature. We annotated roughly 376 articles with about 14 different synthetic biology related organism host and the genetic tools. And then the number of entity we annotated is about roughly 1,800 entity. If you can see here, then there is some E. coli as a host organism and the plasmid IDs and so forth, human manually label. Then here is the annotation status. Based on that, like we can ask a large language model the question. For instance, so if you are looking at here, given the option organism host, promoter, and so forth, which one is the entity type of the J23108? If we ask like that, then this is zero shot. Then the large language model answer. We regularly benchmark different large language model. We constantly doing benchmark. Not only this large language model, protein language model, DNA nucleotide sequence language model, and then the different protein folding and so forth. We are regularly doing it. Here is we are demonstrating on the large language model for this type of the chatbot style. Prompt engineering. If you see the here MPT chat that's showing the roughly macron F1, you can think of it as the micro F1. You can think of it as accuracy, roughly 75% accuracy. Good, but not impressive, right? But it is usable to a certain degree. However, not only just asking the question, let's say if we are providing contextual information where that promoter name was mentioned in the literature text and then provide as additional context, we got the answer with a much higher accuracy. That is so-called one shot. If you see, then we could reach like a 94% accuracy, so much more boost. In other words, providing more contextual information significantly boosts the prediction accuracy. Not only there, and there is nowadays many new concepts in the prompt engineering, like soft prompting, and also like in-context learning. In other words, multiple interaction. I think the Paramvir will show those kind of technique as well. Those kind of many new technique came out. With that, we believe it will going to be much usable and then production deployable. Another thing is that instead of we are manually asking one by one, because we gotta do in the KBase, we need the automatically processing them and then put them into the knowledge base. So can we automatically recognize biological entity and the genetic tool from the raw article text? So we crawled about 71,000 bioarchive article on the these 7 subcategories from the bioarchive, and then we ask the question to large language model, which biological entity and the genetic tools are mentioned in this text, and what are their types. Choose from the following option: plasmid, organism host, promoter, and so forth. And then dump the literature text here. Then it answer with the, let's say, as you mentioned here, organism host, E. coli, promoter, PTIC, and so forth. Then we process those natural language answer into the JSON format, and then it should be ready to load into the knowledge base. Not only that, this is very exciting. Let me try. Can you play the video?
Complementing the development of the large language model to assist research in synthetic biology and biomanufacturing, KBase is prototyping a web application for users to interact with this model through a chatbot interface. Starting with a simple question, we are provided to a demo model that will be replaced by our team's final product. We can ask for information about bacteria and genetic tools of interest, and package the results into a format that populates the resulting user interface. On the left, we see a pruned NCBI taxonomy tree that highlights the organisms of interest in the context of their near relatives. On the top right, this information is summarized for each organism in our query, complete with references. On the bottom right, we include genetic tool information from various data, complementing the development of the large language model to assist research in synthetic biology and biomanufacturing.
Sorry, can you stop?
KBase is prototyping.
Yeah. Sorry. Essentially on the previous slide, we were exploring the model output related to the genetic tools and then the publication reference identified in the literature, so that using those user interface, not just the regular chatbot, but the visualize and interact with the user. And to me, the great value of this interface is that it's all logged. So if we collect this information, this would be a great feature to the KBase service enhancement in the future to me. So here is the team. Thank you.
All right, so I'll be talking about the pipeline that we built in KBase to annotate protein functions using a combination of structure and sequence similarity metrics. And there's a lot of authors and contributors to this talk, including people from the P&L Soil SFA team, people from the U-Biosphere SFA team, the PDB team, Stephen Berkeley, John Gerlt from the EFI team, and I want to particularly recognize Claudia, who did a lot of the groundwork for what I'll be talking about today. So my talk looks a little bit like a yearbook for BER, and I'll try to call out names, but if I miss somebody, your picture's still there, so I'll do my best. So KBase has a 4-step pipeline for annotating functions, for doing functional discovery on the platform. And the first step is to, apply a variety of annotation tools to all the genes in your genome, getting hypotheses for what those functions might be. The second step is to take all that functional annotation information and integrate it to make high-level predictions about what the organism can do. So we're integrating small annotations into high-level phenotypes, like what we have from Dehal's presentation. The third step is to integrate structure-based data to gain more confidence into our annotations and maybe discern whether one gene or another does a function, and I'll explain how that's done in my talk. And the final step is to explore function from an evolutionary context using tools like sequence similarity networks, trees, and gene neighborhoods like we get from the EFI tools. You can look at poster number 48 for more on this. And of course, the fifth step, which you do outside of KBase, is experimental validation, but I want to call it out because it's important and it's not thought about enough. So the first step is aggregating these annotations. And so we've worked with the Lawrence Livermore SFA team, Patrick DeHassler and Jeff Kimberlin here, and as well as the P&L Soil SFA team with Hyun and Amy here, to build tools to integrate annotations, many different annotations from many different tools into a single genome. And then they've added this wonderful compare tool that compares these annotations from all the different algorithms. So each row is a different algorithm. This is all the same genome. And it shows how many functions were annotated by the algorithm and the distinct gene function pairs. And what you see is you've got some algorithms that are far more promiscuous than others. So you've got much bigger numbers in this last column. So I want to particularly call out in this family of tools the PDB annotation tool that we added in collaboration with PDB. So this was a funded effort from the KBase PDB project. This was done in collaboration with Stephen Berkeley. And what this tool does is it blasts all of the genes in a genome against the PDB service and gives you the closest hit to PDB to all the genes in your genome. So you can see how much structural evidence exists for the genes in your genome and how far it is. So you get the sequence similarity of those hits here. And we import a bunch of relevant metadata, including co-crystallization ligands, publications, annotations, PDB IDs, and UniProt IDs. So the nice thing that you get out of this is once you've run this tool, you can click one of those links and instantly go to the PDB function structure page for your particular gene of interest. So immediately bringing structural evidence into the KBase platform. So of course, once we've integrated all these different annotation tools, we're running all these tools, like how good are these annotations? Is there a tool I should trust over another? And so we've begun to evaluate these annotation tools using the KBase platform, and we've evaluated against 16,000 proteins that are in the SwissProt database that have experimental evidence explicitly laid out for them. So these are experimentally validated functions in SwissProt, and you can see the accuracy of the methods. And what you see is, divided by the domain of life. So one thing you see is these different methods perform differently on different domains of life. You also see RAS performs very slightly higher accuracy than DRAM, but DRAM annotates more proteins than RAS does. So that's why there's wisdom of the crowds and applying many of these different algorithms. They have different properties. It's just as valuable in a lot of ways to have an algorithm that has a lot of false positives because it gives you a lot of hypotheses about functions that are useful for explaining phenotypes, which is what we get to in the next step. Before I get to that, I want to talk about an exciting new tool that we're using in KBase to use now LLMs to replace sequence homology. So this has been a constant challenge for us that sequence homology doesn't really scale, but with LLM technology, we replace the challenging sequence alignment comparison process that drives tools like BLAST or any of these other tools that exist today with a simple vector comparison algorithm, which is extremely fast, to the point that we can now get all the nearest neighbors in UniProt with distance metrics in 0.3 seconds for a single protein, which is extremely fast compared to just blasting these things against UniProt. If you've ever run a sequence homology search against UniProt's website, for example. So we're excited about this because this gives us a rapid link to UniProt, which gives us a rapid link to AlphaDB. So this is there'll eventually be an app that comes out of this that will annotate a genome rapidly with this tool with UniProt, and it will automatically bring in AlphaDB structures for those UniProt hits when the user requests them. So it's we'll have some nice ability to pipe into that AlphaDB resource. So going on to step 2, now we want to integrate experimental phenotype data and attempt to use our annotations to explain that data using different tools like modeling and machine learning. Towards that end, we've integrated a bunch of phenotype data. This is done. Paramvir is involved in this. Ranjan is involved in this. Zamith, the postdoc in my lab, and Jim Davis and Pam Weisenhorn contributed some of this data. We've integrated a bunch of different data sources for phenotype data, and we've applied, this is one use of this data is to evaluate our annotation tools. So we can build models from different annotation tools and then evaluate their accuracy based on how well these models perform and predicting the phenotype data. So we can mix and match as well. So we can build models from multiple annotation tools, and you get different properties from these models in terms of different numbers of false positives and false negatives. We can also build models from machine learning, which has an advantage over mechanistic models because you can use all of the features, not just the features that are involved in the mechanism. And for that reason, we actually get a slightly higher performance from machine learning versus our metabolic models, and we can fit our models to the machine learning and further enhance accuracy. So this is something we're exploring, and as our machine learning tools get better, and it's really driven by work that Paramvir is leading right now, I think this capability will improve. We can also fit the experimental data directly, so if you have an isolate with experimental data, as the PNNL Soil SFA team did, they have 7 isolates with experimental data for these groups. We can use the modeling tools, specifically the Omega tool that we co-developed with the PNNL Soil SFA team, to fit the models to the tools. So what we do is we build the model initially from the highest confidence annotations. And then we use all those other annotations from all those other tools as we cherry pick from their annotations such that we can explain our phenotypes. So not only do we fill gaps in our model when we fit the model to the experimental data, but we also find gene candidates for those gaps. We're able to propose gene candidates from those other annotation sources that we didn't include in our initial model. So this gives us an ability to flexibly use annotations, and it's all because of the probability work that the LLNL team has done and all the work that's been done to integrate all these different annotation tools. We can also predict novel reactions, and for example, using the pickaxe tool that was co-developed with Keith Tayo and Linda Broadbell at Northwestern University, and we've applied these tools to predict novel degradation pathways for pyridine. The left pathway has actually been experimentally validated fully, and the right pathway we're still validating, and we can predict functions or genes for these novel steps based on functional similarity. So if a novel reaction is similar in terms of the chemical structures that it operates on or the chemical transformation that it performs, then we can say, oh, this gene that's annotated with this 1.14.13 EC level could probably also do this 1.13.14 novel reaction. That becomes a gene candidate. But now this is a weak candidate, right? We're proposing a totally novel reaction for an existing gene that's not explicitly annotated with that reaction. How do we improve our confidence? This is one place where structure can come in. So we can fold the structure outside of KBase with AlphaFold, import the structure into KBase, and then dock the structure using docking information from existing co-crystallized ligands in PDB, and using the AutoDock Vina, that tool that was wrapped by Ada Sedova, and then gain further confidence in our annotation based off of docking scores. And this is important to get context here. So we have to run many different derivatives of our structure and many different protein candidates in this tool, so we can get many docking scores and just confirm that the score from our protein candidate stands out among all of the others as a likely function that this protein will perform. Once we've done that structural validation, we have some confidence in our tool, we can go to our evolutionary approaches. So here we take our VAN-A gene that we think does this monooxygenase step in pyridine degradation, and we first build these, we submit this to the new sequence similarity network tool that's now being added into KBase, should be available in beta in the next week or two. And this tool will build what's called a sequence similarity network, where the nodes are proteins and the edges are whether or not those proteins are homologous to one another. You start with a hairball because it's got a very a conservative prediction of homology, but as you increase the threshold of homology that you have to have an edge, you lose edges in the network. And as you lose edges in the network, you can untangle the hairball. In the same way that as you have less hairs in a hairball, you can tease the hairball apart and start to untangle it. So that's what I'm doing here, increasing the threshold, removing edges, increasing the threshold, removing edges, and starting to get these fine-tuned clusters that are small enough now that I can rapidly build trees, and I can iterate between these two tools. So trees aren't in the EFI tools, they're in KBase. So that's one of the things we're doing in this effort, is integrating these tools together so they work in concert to give you a very fine-tuned understanding of what proteins are isofunctional and what functions to assign to each of these subclusters. The EFI tools also offer a gene neighborhood tool, so we can see if these isofunctional clusters have a conserved gene neighborhood and if the function we've assigned makes sense considering the neighbors that we find in that conserved gene neighborhood. So in this case, it does. We see a conserved neighbor, which is an essential part of the complex for doing monooxygenase. So it makes sense that this VAN-A would do this function. I want to talk about ukes a little bit because I don't want to have a totally proc story, but we can also do this in fungi. In this case, we took 13 published fungal metabolic models. We have a poster on this, number 66. And we loaded into KBase, and the first thing we did was to make the chemistry of the models consistent. They all had different representations for all the pathways. It was a mess. We made it all consistent. The core is totally consistent now. We now have a consistent metabolic core for all fungi. And then we're working on the periphery at the moment. And what this lets us do, once the chemistry is consistent, we can then build protein families and see if the annotations are consistent. And what we find is they're not. Every yellow bar here represents a case where a model should have had a function and it doesn't. And this is the most important metabolism in the model, and they still have omissions. So we use this approach to find trouble spots in the fungal annotations, like aconitases. So how do we deal with these trouble spots? Well, one thing we can do is look at structure, and specifically we took two very similar aconitases. This is citrate dehydratase and 2-methyl citrate dehydratase, two very similar structures. And the first question is, can structure help me discern these things? And what we see is yes, these two functions, so if I take the genes associated with these functions in yeast and E. coli, I find that the structural homology is very low between citrate dehydratase and 2-methyl citrate dehydratase. This tells us that I can use structural homology to discern this function, which then takes us to the next step. Now I can look to see if the yeast citrate dehydratase aligns with the E. coli citrate dehydratase, which it does, and I can see if the 2-methyl citrate dehydratase aligns with the E. coli 2-methyl citrate dehydratase. This is an example of using structure to propagate function instead of sequence. In the case where sequence fails because the homology is just too high. This worked successfully, although we were more confident about one than the other, and one should be experimentally validated. So then we can use our EFI tools, build the EFI network, again tease it apart, and assign specific econotate functions to every subcluster in this tree, and they each have their own function. You can see the clusters from the coloring are very consistent. You get the same clusters from the EFI as you get from the tree, and that we can do a much better job of annotating this function across all the fungal genomes. So with that, I want to talk briefly about some work we have going on in the experimental validation. So we're now developing a self-driven lab at Argonne National Lab where we're collaborating with Ellen Needle to implement her ADP1 natural competence-based transformation system so you can rapidly do we're implementing an automated complementation and knockout protocol for ADP1 that we can implement to, for example, complement a candidate gene that has a function and a strain that has that function knocked out and test to see if the strain then grows. With Ellen's protocol, we can robustly express that function and get activity even if the sequence isn't ideal for the particular strain environment. So this is an active thing that's being funded by LDRD now, and we hope to get support for this in the future. And with that, I'm going to recognize the KBase team and the many, many, many teams that contributed to this effort and the funding sources, the SFA Plus Ups, KBase PDB and KBase EFI, and the KBase project itself.
Hi, good afternoon. My name is Bill Nelson. I'm a scientist at PNNL. And today I'm going to talk to you about some of the tools that we have been developing that will hopefully help with the gap filling as integrating various data types that will help provide functions that we can use for gap filling in metabolic models. Okay, so this work has been largely developed under the two SFAs that we have that are funded by DOE, the Soil Microbiome SFA with the PI of Kirsten Hofmackel, and the Persistence Control SFA, PI is Robert Egbert. And for the Soil Microbiome SFA, what we're trying to do is use a multi-omics approach to gain a predictive understanding of soil microbiome interaction with a tall wheatgrass that we have growing in the field, particularly under drought conditions, and how those interactions mediate carbon flux through the soil system. And in the Persistence Control SFA, we are looking at how to engineer organisms to controllably integrate them into environmental rhizosphere communities. Again, interactions with plants are going to be important in that. And so in both of these, we're collecting multi-omic data, and we are looking at interactions between organisms, and we are modeling the activity of individual organisms, which means we're doing metabolic modeling. And we're running into that age-old problem that everyone who does metabolic modeling has, which has been mentioned here already, which is gaps in the models. Gaps continue to be a significant issue in metabolic modeling, and this is basically because we don't know all of biochemistry. We've been doing biology for a long time, but apparently there's still some stuff we need to learn. There are a lot of genes with completely unknown function, or at least poorly described function, that you can't explicitly map within a metabolic model. And we also have just nutrient or metabolite transformations that we may observe in an experiment, but we don't know the biochemistry behind those. We don't know the genes involved. We don't even necessarily know the transformations involved. And so these wind up being gaps in the metabolic models that we build. And the current methods for gap filling models tend to fall into two categories, one of which is just, if you have a growth condition that your model is supposed to operate under and it doesn't, then we'll just assume that the missing reactions are present. That's one method. And another one is you can always lower the functional classification thresholds and see if something will pop up that you can fill in for that. But these tend to be parsimonious methods that can lead to models that are not as predictive as we would like them to be. So we've been collecting as a community, not just us, as a community, we have been collecting a lot of genomic and metagenomic and experimental data for a long period of time. So now can we apply some machine learning or artificial intelligence techniques to this extensive and growing body of information to improve the accuracy and extent of modeling and gap filling. So I'm going to talk about three tools today, pretty briefly, and then also a brief vignette for each one explaining why it may be relevant to gap filling. And then we'll wind up tying them all together at the end. Okay, The first tool I'm going to talk about is MetaPath Predict. This is out in a preprint right now. It's largely the graduate work of David Geller-McGrath up there in the corner there. He's a graduate student at Huey under Virginia Edgecombe, but he was collaborating with Jason McDermott and Travis Wheeler to build this tool, which what it does is it predicts complete KEGG modules from incomplete KEGG ortholog data. And I imagine everyone in the room is familiar with KEGG orthologs and KEGG modules, but for those of you who are not, a KEGG ortholog is essentially a gene function, a specific protein function. KEGG modules are when you stitch those together into pathways, so this is more of an indication of the cellular function or a phenotype, perhaps. And MetaPath Predict takes a stacked ensemble approach, combining neural network and gradient boosted tree predictions, combining those together to come up with a final prediction. And what we did to train this MetaPath Predict was we had a collection of 24,000 complete genomes that we used. Those were run through KEGG annotation, and then we chose 293 KEGG modules that were complete in at least 5% of those training set genomes. Those were what we were going to predict. And then these were, the final models were tested on a held out set of genomes, I believe it was around 6,000, something like that, for which they were, again, were run through KEGG annotation, but then we subset out just certain subsets of the KEGG ortholog information to run the predictions. Okay. And so here's the result, presenting an F1 score, this combination of sensitivity and specificity. And this is in comparison with some other modeling techniques, just a gradient boost or a multiparametric linear program, or those two, an ensemble versus the MetaPath Predict. And what you can see, that this is on the x-axis, this is the percentage of KEGG annotations that we included in the test set. So going from 90% all the way down to 10% of the data, so really limiting the input information. And you can see that not only did MetaPath Predict perform quite remarkably, but even under very little input data, it was pretty accurate. So so this is a great, you can see how this might be applicable to if you have an incomplete genome, if you've got a MAG coming out of a metagenome, or a draft genome, or even single cell rolling circle replication genome where you think you don't have complete information. This will allow you to predict functions that may be present in your genome, and that will help you build your model. Okay. The second tool is Omega, and that stands for Omics-Enabled Global Gap Filling. And this was primarily developed by Hyun Song up there in the corner. And this is a gap filling tool, which is now available in KBase. I believe it's almost fully functional there. You can use it now. It was presented in the KBase workshop yesterday. And the idea behind Omega is that it allows you to do global gap filling, simultaneous global gap filling. So if you have multiple growth conditions that you want to gap fill your model against, previously the way you would do it is you would iteratively, gap fill against growth condition 1, gap fill against growth condition 2. And we have found that this leads to models that are not as accurate as we would like them to be. So Hyun came up with an approach to do simultaneous global gap filling to take care of all of that at once. In addition to that, you can also do omics integration. So if you have experimental transcriptomic, proteomic, or metabolomic data, that data can be used to parameterize the template model that you're drawing reactions from to ensure that flux gets pushed through reactions that you're detecting to improve your gap-filled output model so that it is more biologically relevant and more predictive. Okay, finally, we have SNECMR, and this is our evolving annotation tool, which we've been working on with Jason McDermott, and Christine and Jeremy and Tara have been doing a lot of the actual on-the-ground programming for that. And this predicts functions for proteins using a concept where we're looking at recoding the protein sequences to smaller alphabets to both simplify the computational problem, but also to essentially take into account both observed and unobserved sequence diversity for the protein families. And then we use a K-mer profile analysis to actually do the comparison between proteins, because that has computational, it just works better computationally. And this tool has also been published and also is available now in KBase. There's a couple of different libraries of ontologies that you can run your genome against using SNECMer. And so what I'm going to show here is trying to convince you that this concept that I said about having unobserved diversity be accounted for in our models. So what we did was we took 1,000 complete genomes and then we dropped out different entire clades to do model building. So for instance here, we dropped out all of the gamma proteobacteria and then built models against everything, and then the dropout set, and then tested against specifically the proteobacteria, the gamma proteobacteria. And we did this for all these different clades. And you can see that there was a pretty modest or even no drop in accuracy for the identification, the classification coming out of the SNECMer models. So hopefully this convinces you that if you happen to have a protein that is more highly diverse or an organism that's more highly diverse, SNECMer models should be able to give you a reasonable annotation based on that. So now let's look forward a little bit and say, well, how can all these things help with gap filling when mixed all together? So if you start with your either complete or incomplete genome, you can use SNECMer and many other tools to get your annotation and build a template model. You can use something like MetaPathPredict to also help you fill in information about functions that you are predicting to be present. And all of this can be input to Omega, where you can also include your growth data or your multi-omics data, or maybe even, we haven't done this yet, but it would be nice to be able to integrate some of the fitness data, the high throughput fitness data that's coming from RBT and seq and CRISPR-I experiments. And Omega will run its magic and we will get supermodel out over here, which should be more biologically relevant and more predictive. I had two ways I could go with supermodel. I decided superhero. Okay. So that is my talk. I would like to thank all the people on this slide here, the people at, oops, not that one. People at PNL, Woods Hole, University of Nebraska, University of Arizona, and call out our KBase collaborators, Chris and Janica and Elisha, for the help they've had in getting these things integrated into KBase, and the Department of Energy and the NIH for funding. Thank you.
Questions for Bill?
Yeah, that was great. I'm familiar with methods that use phenotype data for gap filling, so like biolog type data. And it's most of what I've seen uses kind of the positive growth substrates. But there's a bunch of data that's just sort of thrown out, which is all the negative conditions. So I'm curious if you guys have put any thought into what to do with that, that there might be, that your draft model might be able to grow on a substrate that is then, not in the biolog data.
Yeah, we actually have observed that. We have done gap filling on models and then compared it against growth data that we have, and we have seen false positives where a model will grow. And usually that requires some tracking down, trying to figure out, I think for one of them we figured out it was unbalanced reactions that were causing a problem within the model. Chris might be able to speak a little more directly to that. But that is something that, it is another form of error in the model that we should be able to solve using proper gap filling, a true understanding of what the functionalities that are present in the organism.
Yeah, it's something we've given a lot of thought to, and that we've actually corrected the gap filling algorithm to have tests on any reaction that's considered for inclusion in a model And any reaction that, for example, breaks mass balances or breaks ATP production is stripped out of the database. But we also have the ability, I don't know if this is implemented in the production tool right now, it might be, but we have the ability to say, for the user to say, I don't want a model that grows on these conditions. So make these conditions work, don't make these conditions work. And it will go on to also exclude any reaction that causes those conditions to work. But makes the other conditions work. We don't really have a tool for stripping stuff out that's already in the model that's causing a false positive. And the problem there is it's just, I mean, there's so many things you could pull out, and there's the obvious solution of just stripping out the transporter. So, that's the tricky thing. But we definitely can prevent you from creating new false positives when you gap fill your model, which is a huge step forward, I think.
There's a question over here.
This is how I get my steps today.
Probably I should have a question for both of you, but yeah, both great talks. So I'm seeing all these like amazing new kind of like function prediction, module prediction methods. I would love it if there was some way we could get to a kind of like a leaderboard, that like a Kaggle or Hugging Face one, where I could just see, okay, for fungi, for kind of archaea, for this task, which is the best one to use. But that's really challenging, because how do you evaluate it? We heard from Chris, for the function prediction, you evaluate against SwissProt, but that's going to be highly incomplete, and that won't give you a way of evaluating against, predicting whole kind of like KAG modules. So it's a bit kind of like, broad in scope, but what are your thoughts as to getting towards that kind of leaderboard?
So ever since my days of annotating genomes back at Tiger, my opinion has always been more information is better. So I really like the project that Chris talked about, that Patrick was working on, where, how can we take many different forms of annotation and integrate them and try and figure out which ones have the highest confidence, which ones seem to make the most sense. So I am all for it. If there's a new tool out there that seems cool, then go ahead and run it and integrate that into your analysis. We'll figure out. I think that's sort of a next step, is continuing on this path of how do we integrate across annotation types that may be more or less accurate for either different organisms or different types of protein.
Yes, I think our thinking along that lines really dials into the talk that Adam gave about this CDM. So imagine a central data model that has all of the RBT and seq data. It has all of the phenotype data from all of the SFAs. It has all of the gene neighborhoods. It has the trees. It has all of the ontologies perfectly mapped to go with reactions, with good high quality reactions mapped. And so anytime you make a new annotation, you can run it against all that and get a consistency score, like, you're really consistent with gene neighborhood, and you're really consistent with structure. Like, that's the goal, is to really connect. We have all that data. It's just too siloed right now. If we can connect it together and integrate it properly, it gets really powerful.
All right, so I'm gonna talk today a little bit about some of the work that we're doing with LLMs. We've got a lot of projects, but I want to focus mainly on one of the things that's kind of nearing completion and hopefully can be released soon. But you saw from Rick's talk yesterday, he spoke about these ideas of agents that would help act on the user's behalf. And this is one of the things that we've also been very interested in. And so, he was talking about like these very large agents that would just kind of do everything, do your Zoom meetings, text chat, but take high-level instructions and then work towards that goal. And that's the same concept that we have. Within KBase, we have a very kind of confined, constrained environment. There's something like 500 plus apps. I don't know how many hundreds of data types. But this is pretty overwhelming of like how to decide what to use, how to use it, how to put things together. And it takes a lot of users' time to figure out how to use those tools properly, what parameters to set, what parameters matter for the type of analysis you're trying to do. A set of parameters that are appropriate for one question may not be appropriate for another question. And it's very complicated to figure that out. So what we're hoping to do is have a KBase research assistant that's capable of taking the user's goal, devising an analysis plan, and then actually executing that plan on the user's behalf, summarizing their results, and then providing the kind of classic KBase narrative back to them. So as a sort of proof of concept for this, we decided to narrow the question a little bit down to just writing a genome paper for a user. The reason we picked genome paper is that the process for writing a genome paper is reasonably well constrained, and there are a lot of examples of genome papers. So we were pretty confident we would be able to do that, but then, open it up and do more. So there's a lot of need for this, and I think we're trying to follow a lot of our kind of KBase principles here as well, where we want to make sure the data is private, allow the user to kind of explore and process it before they have to make it accessible to the public. So we're not trying to automatically do this for everybody. And the research assistant has to perform kind of tailored analysis on the user's data, construct custom workflows, interpret the output of one step in order to parameterize the next step. And then finally, we want to actually help the user publish their data. So I don't know how many projects are just sort of sitting on thousands of unpublished genomes because they think maybe one day I'll actually write a genome paper about it. Hopefully we'll be able to shrink that time and let the user just kind of quickly iterate, answer questions, work with the research assistant, and get these genomes kind of out the door so that everyone can use them in their analyses. So where we started was the first thing we did was we just made like a chatbot for user analysis, where we took all of the documentation that KBase has made over however many, 11, 12 years, which turns out to be quite extensive. We've got a lot of documentation. We've got a lot of tutorials. I don't have this one called out, but one additional source of training data that we have is the material that Ellen Dow put together for the KBase Educators project. And it turns out that the same kind of instructions that you give undergrads will work for these language models. So that was very fortuitous. So we take all that documentation, make embeddings, put it in a vector store, so that whenever someone comes in, they make a query, and this is also trained on some of our help desk. You could basically just ask it any question, and then it would go back, retrieve all the relevant information, and then give you an answer. And I think that we were all pretty impressed by this chatbot. And so instead of like just stopping and saying, hey, maybe we should release this and let users actually use it, we thought, oh wow, this is awesome. Now let's actually have it run the analysis for me, which I think we should have published this first. Because it actually does take a little bit more time to make an agent. But we got excited. But the other thing I wanted to point out is that we did use a lot of different models. We've kind of focused on using the OpenAI GPT-4 as our kind of rapid prototyping, and then we have some of the open models that we can do some fine tuning on, as well as like just keep it all in house, because ultimately we don't want to use something like GPT-4 where it would require your data getting sent out, and then who knows how they would actually use it. So, as I said, we kind of focused on the genome paper, and fortunately for us, Zach has put in a lot of work working with the MRA microbial resource announcements publication, to get them to accept effectively KBase narratives as part of their genome paper. So if you do your analysis in KBase following this template that he's produced, it'll, I don't know, accelerate the review. I can't remember exactly. So we started with this, making sure we kind of trained on how to do it, and then so we took the template, and then we also took all of the genome papers that were written in the MRA format through KBase, or using KBase, and most of this came from some of the work that Ellen did with the KBase educators. So she's got plenty of papers that they've put together. And so how does this work for the user? So first they kind of come in, they write down their goal. In this case, it would be writing their genome paper. And then the agent gives them a suggestion of what to do, and the user has to approve it before it actually runs. And then the agent will run it for the user, and then summarize those results, putting them in the narrative, and then recommend the next step. So this cycle kind of repeats, where the user has to approve each step. And we kind of put it in this way as sort of like a safety feature. We want to make sure that the user is aware of what's happening, and they decide that it's the appropriate thing to do. And then at the end, the agent does the writing. And it turns out the writing is the easiest part. We've got the template, you just put in a couple blanks, and then the agent just writes it. It's kind of amazing that it just worked and we didn't need to do any fine tuning. So it's a little bit different on the agent side. So what we do is we have several agents that are working together, and so when it's working, it gets the request from the analyst agent, starts everything. It gets the request, figures out what needs to be done. And then once it decides what has to happen, it talks to the narrative agent. And the narrative agent is the one who knows what our KBase narrative is, knows what the rules are, knows how it's supposed to work, how to invoke apps, how to read the output. And so it tells the narrative agent to run the app for it. And then the analyst agent analyzes the results again. So if you ran something like, the first step is probably something like FastQC, the analyst agent then kind of reads the results, interprets the results, and then tells the user what the summary is. So it could say, one of the things it can say is like, you've got a lot of adapter. I suggest you run Traumatic and clip off your adapter. If it's not one of these in the dropdown, just use, a clipping score or a pre-clip of 5. It's very detailed parameter instructions where the user may not have even known, someone told me to run FastQC, so I ran it. I didn't really understand, so then I just went to the next step. The analyst actually helps you with that. And it runs all of these things automatically. The one thing I want to say is that we don't have that many agents, and each agent has a very specific role. We can explore having additional agents. One typical thing is to have an agent that's like a critic that would, once a step is proposed, the critic, criticizes it and says why it's a bad idea. But the problem we've had with that is you kind of get stuck where sometimes the agents start fighting with each other. And so for this purpose, we just decided we would start simple and just have the user be the person who decides that it's a good idea or not a good idea. If we had the critic, you can imagine taking the user a little bit out of it and letting them run through. So, and then at the end, the writer agent writes it. One of the other aspects of this is that we have to have a lot of tools for the agents to use. So the agents are trained on some information, they know what to do, but they need to use tools to do them. And for within KBase, obviously this means we need to have tools for interacting with our narratives, interacting with our workspace, interacting with the data. But there are a lot of other tools that we could have. In fact, there are several pre-built tools, but we haven't brought those ones in yet. For example, often what you'll want to do when you're at the like a writing step is you'll actually want to kind of go out to the literature and see, what are common themes about the genus or species that I have my genome for. And, there's like Wikipedia tools. Those haven't been integrated in, but those are things that we're looking at to let the user bring in more information. And then, so the agents all have very specific roles. And so they have these capabilities of doing reasoning. They remember what happened, what you said at previous steps, and why you want to do what you're doing. So there's a lot of kind of debate over whether or not LLMs or these AIs actually reason or not. But I'll just say for the purposes of what we're doing, it sure looks good enough, like good enough for our purpose. So whether it's real or they're just pretending, it's fine. And so we've started a lot of this with retrieval augmented generation, and so that means we can quickly bring our documentation, put it in. But one of the although RAG does help in kind of preventing some of the hallucination, it still has like some problems where you'll ask it to do something and you'll tell it that you want to do it and it wants to help you. So it'll suggest like an app that KBase doesn't actually have. And when it does it, you read the app name and you're like, yeah, KBase should totally have that app. It's like, that's a great idea. So in order to get that to kind of stop, we've, this is just a little bit more detail on how the RAG works. So in order to get around that, what we did is implemented a knowledge graph. So instead of just going to like the App Store, we've created a knowledge graph that links apps to the data types that they operate on and to the, again, the apps that operate on those data types. So there's only a limited number of paths through it, but still 500 apps, hundreds of data types, there's lots of paths. But what it means is that when the agent decides that it has to do some function, it'll query the knowledge graph and figure out like what the appropriate tool is, and then it'll only use that tool. And so it knows what it can do immediately, and it knows what it can do next. And so, effectively this is all stored in Neo4j, and it turns into a Cypher query. It's only relevant because later we'll probably want to use the same thing when we augment the graph to have more data. Right now it's just focused on tools, but there's no reason we can't augment it to query something like the CDM that Adam spoke about. So this tool was implemented, and so the next steps, so we were actually kind of hoping we would get this done before this meeting, but there's several steps that still have to happen. Primarily, we really haven't focused on the guardrails and the safety. We've designed the tools such that the agents can't necessarily do malicious things to your workspace, because there's only so many things they can do, and deleting or sharing your data is not one of them. But we still need to kind of figure out all of the guardrails. We're trying to follow as closely as possible the White House guidelines that came out in October. I think there's been some updates to that as well. And then we need to do some more extensive user testing. And then after that, so right now a lot of the user testing has just been within our group, and our benchmarks have just been very limited to the writing genomes in a best case scenario, not like what happens when things don't go well. So we need to improve our benchmarks and our user testing, and then we want to add more data to the knowledge graph so that the user can actually ask more complex questions, and then add in those tools that allow the user to go outside, collect some papers, and then improve the writing of their paper. And with that, I should have said, so currently this is all functional, but it only works in a Jupyter notebook, and Prachi's currently working on getting the UI up for it. So I want to thank the KBase team.
Question for Paramvir. Chris, you already got one, so I'm going to give it to her.
So just wondering if you have socialized this idea with scientific journals and see what their requirements would be for such publications?
Yeah, so we haven't reached out to, so that's a good question. That is definitely something we need to do. A lot of the, I think there's like a split. Like one group of journals have basically taken the position that as the author, you're responsible for the content, and the author should figure out how they want to use the text generation. So if they're just using it to kind of improve things, or if it's being used to kind of come up with the ideas, those are different use cases, but still the author is responsible. And then I think there's other journals that have just been more reluctant and said, yeah, you have to write it all on your own. But yeah, we need to reach out to MRA and just let them know what we're.
I've done that, and I got the "we are evaluating our AI policy" answer. Please come back to us later. Okay, one more quick question.
Okay, this is all terrifying, but also super important. I'm imagining a scenario where this assistant is deciding to provide guidance on a particular step in a long series of steps, and there may be a multitude of tools that can fulfill that step. They're available in the literature, and it has to choose from among them. How do you imagine that these choices will be made? How will it deal with issues such as recency? You don't want to suggest that somebody should use FRAP for assembly, for example. And is there a notion for handling kind of confidence in choice or representing confidence in choice?
Yeah, so that's a really good question. And one thing that's kind of complicated and a little, since we are building off of a foundation model that's trained on some corpus of information that we don't necessarily control, we only control like that fine tuning that we provide on top of it, or how we do the biasing in the RAG step. So surprisingly, it's actually pretty good at recommending the appropriate app. Like I'll use assembly as an example. It understands that when one tool is appropriate for another, and it'll tell you what's available. So it'll say something like you can use Metaspades or IDVUA, and then it'll tell you that Metaspades is the one that most people use, which is right. And for most people, that's probably what they should start with. But the other thing that it does is it just lets you run multiple assemblers, and then it interprets the assembly output and tells you which one's better. So we don't have computer vision, but maybe we'll have access to a model that has computer vision so that it could read the graphs. But it can read the table, the tabular data just fine, and then it'll say like this one's got the best N50, has the most bases. or for whatever reason this one had a very fragmented assembly, and it'll give you an explanation of why it thinks one is better than the other. But as far as like recency bias, I think a lot of that will come down to our training data. And so if we're using things that are more common or more recent, or just what users are doing. And then the other thing is that because we're pulling the apps out of the knowledge graph, so it's not, it's limited there, we can encode a lot of information inside there about like what the errors are, where it's appropriate, where it's not, and that gives it more information to make that decision, or at least give that information back to the user to help them make that choice.
Okay, I'm going to give the last talk, and I recognize we're short on time, so I will go through this relatively quickly. I wanted to wrap this session up with a little bit of a reassuring message that a lot of the data integration talks that you saw today and the session before this really involve a lot of people coming together with a bunch of different kinds of data. And so what I wanted to talk through at the end of this was just understanding how to make sure that you can still get credit for your contributions in a big data world. So science is a creative pursuit. It takes a lot of creativity and ingenuity to be able to put together a good research question, to be able to design a quality experiment, to combine data in new ways, to discover novel patterns, and then to actually craft that language to share and extend that. There's obviously a lot of publicity right now around AI stealing from various different groups, being musicians or artists, and so I think that there is something to be considered here, and we want to make sure that our careers that are all still built on publishing and getting credit, how do we actually share data into this data integration space and still be successful? So you probably have all seen this at some point. The year of 2023, last year, was declared the year of open science. The federal agencies all signed on to it. OSDP put out a memo in August of 2022. And then DOE has put together a public access plan that was open for comment. And the main thing that they're really looking for is making sure that the data behind the publications is free, is immediately available, and that there is a persistent identifier assigned to that. And I'm going to use this talk to explain the importance of that. Leanne also talked about that in terms of the standards conversation around the NMDC stuff. We are working on the back end to make sure that all of your contributions are able to be tracked, not just the publication. So this was an interesting article that came out a couple of years ago that talked about why researchers don't share. The three main categories are disincentives, so things like being scooped, not having enough time, knowledge barriers, my data is really complex, I don't know where to put it, maybe there's an unclear process at each of your institutions or by your funding agency, and then reuse concerns about it not being used appropriately, not understanding how it was generated, or what the sharing or licensing is for it. So this is how we are all familiar with tracking research products. You write a publication, that publication gets a DOI, and somewhere in that publication you might mention the software you used, where your samples came from, any data analysis that you might have done. Maybe there's some place that you put a data set that maybe somebody can ask for it. And so this is how we've all been publishing in the past. Now there's things like natural language processing that can extract some of this, but still it's a developing process. And so what we're trying to move forward with is an actual workflow where you, as an individual person, are part of an organization. You submit a proposal that gets funded. That proposal hopefully has a data management plan. Everyone in this room should know what a DMP stands for by now. And in that data management plan, you talk about your sampling process, your protocols for processing those samples into data, what kind of data you're planning to collect, what software you might be using to analyze those data, and then of course that the end result is the publication. Well, guess what? Now there's persistent identifiers for basically every step in this process. All of the persons now can get an ORCID, so it's a research identifier ID. I'm assuming that most people in this room have one. If you don't have one, go get one. You have an organization that probably has a research organization registry. The funding agency also has a research organization registry. So that gives credit to the organizations that you belong to and that fund your work for the proposal that they're going to give you money to do. That proposal or that data management plan might have a DOI, and then that DOI can get linked to all of the research products that come out. I'll show an example of how this is all coming together in a minute. So this is the base of FAIR, right? This is making things findable, accessible, interoperable, and reusable. For example, we talked a lot about these various different data platforms that the BER funds. NMDC has a lot of sample metadata and standardized data products. JGI produces a lot of sequence information. EMSL produces proteomics and metabolomics. And ESS Dive carries a lot of the biogeochemistry measurements coming from our friends in the environmental side of the camp. And so if we were able to actually go and put a data transfer service together where you could pull all of that information from a basic search query, so you no longer had to download and re-upload, you basically had a platform agnostic, API centric way of doing this, and you didn't have to go to each individual platform itself. And then we're calling it frictionless. It brings over the data package as well as an informational package or the metadata that describes the credit information. So who funded it, who owns it, what organization do they belong to, any identifiers that come with it, etc. So there's no loss of credit or attribution. The persistent identifiers are all maintained, and the schema is compatible with the publishing infrastructure. So if you bring that into KBase, for example, and we're piloting this with the JGI KBase connection to start, if you bring that into KBase and then you go and publish with KBase, we can actually send all of that citation information to the publishers. So what does this look like? So Michaela Borton is here. She's an early career researcher. She runs the Genome Resolved Open Watershed project. She has an ORCID. She works at Colorado State. They have Aurora. She has a project that was funded by Department of Energy Biological and Environmental Research that then got a JGI CSP, which has a proposal identifier. She then went and collected samples. Those samples have identifiers. She used protocols to process the sample, turn it into a data fair narrative, which I'll show you in a little bit, with software that's also in KBase that's published. It's KBDRAM, and then has a bioarchive article with the persistent identifier. That'll get updated because we're resubmitting this, the revision's going into Nature. And so this is an early career researcher who's doing open science, who's sharing her data prior to publication, and she's doing it in a way that we're doing our best to make sure that we're supporting her so that all of the information that she's sharing gets the citations that it deserves. This is not just, obviously, the JGI CSP, right? She's leveraging a lot of these different portals that are all funded by BER, and so this is a slide from Joe Damerow at ESS Dive, working to actually connect all of these different components and the relationship types between them. There's a lot of information on here, but basically this gets encoded in a JSON schema, and this is then interoperable with the publishing schemas as well. And then in KBase, so Kayla brings this stuff in. Kayla's now the orange researcher, just for the record. Kayla brings her data in. We recombine it with other data. She publishes that fair narrative. She gets a DOI. Someone says, oh, there's really cool river data here. I also have samples from this river. Let me add my data to it and then reprocess some of it. And now I'm going to publish. And so automatically, when they go and publish their blue fair narrative, it automatically cites the orange narrative. And we can tell Kayla, someone's been using your data. And they have another publication out of it. So in KBase, the provenance of all of the data in KBase is integral to the platform. And so we're very effectively leveraging things like fair, trackable, persistent identifiers to then report back to people, this is the impact your research is having. It's also nice because of the reproducible notebook that Paramvir had talked about, allows people to actually understand the data better, and that helps people really understand the context and how to reuse it effectively, and also where it was generated and who generated it. This is what it looks like, for example, of one of our users. So this is a group out of Brazil. They have a fair narrative where they used 16 KBase apps to process a bunch of metagenome assembled genomes, have a couple of publications related to it about microbial functional diversity in Amazonian soils. There's a link to the KBase narrative there and the DOI that's associated with it. Then I can report back to them that their fair narrative, the one on the right over here, 202 people have viewed this. So this is just a general Google Analytics number that we can report to them. But then inside KBase, I can say, oh, and 6 unique users, people that weren't associated with the study, have gone and created 17 new narratives using data from your analysis, and they have 3 new derived data objects. I don't know what those data objects are because they're private, but it's new knowledge that's been generated because someone took their data and did something else novel with it. And to Paramvir's point, we can train the next generation, both of undergraduates and of AI. So this is basically a group of undergraduate students that Ellen has been helping, and they publish these ASM MRAs. Paramvir already talked about the template to go through, and we've talked a lot about the vast number of isolates. And so if anyone in this room has isolates and are interested in going through a mentoring process to train the next generation of students, we have a playbook where we're happy to work with you to get those isolates into the students' hands. They then work their way through the process, and it's a little bit of a mentoring relationship where you explain the experimental design and the samples collected, and at the end of the day, you get a co-authored publication with these students. So let me know if you're interested in that, and we'd be happy to talk more. And that's it. And this, you've seen this picture of the KBase team like 5 times now. Okay, any questions? Final questions, otherwise you are released. Yes. Oh, dang, I have to do this too. Well, the online people.
I guess I'm just wondering, do you anticipate a future where we leave the journals out of this loop? Because, I mean, they're really the ones profiting from this in a lot of ways. And they're also the ones slowing it down.
Yeah, so we've actually talked quite extensively with the ASM Microbiology Resource Announcement editorial group. And this is being recorded. Okay, well, don't quote me on this too much, but we told them about the template.
It's not me asking the question.
No, it's not Mitch asking the question. And so we've actually shown them the narrative that KBase is using to train the AI, and it was their idea, like how do we put hooks into that to auto-generate a PDF, basically. And so we're moving in that direction. We are going to be formalizing a KBase collection with them, so all of the publications that are going to be coming out, and we're actually training the editors to help review some of those faster. So I've warned them that we have an education program, and they're starting to get big chunks of these kind of at the end of the year as the students get ready to publish. And so we're trying to accelerate that review process as much as possible. But ideally, we'll just basically push a button. They'll print out a PDF of this, and then we'll be done.
Thank you.
Other questions?
I can speak loud.
Well, there's online people.
Yeah.
Now I get my steps into
I'm going to make Mitch run.
Well, just on this, I mean, my question on like how like it feels like, and it's also kind of a shout out to you, Elisha, like how many people like are involved like in this, like there's so many connections, like how many, like do you have like a rough estimate of how many institutions, people like you were coordinating to make this happen? Because I mean, you just show like a picture and it looks like simple, but it feels like there's a lot of people involved and that you've been putting a huge amount of work into this.
Which part are you talking about?
When you go to that, go back here.
Oh yeah, no, this is a multi-institutional effort. So Joan Demereau and I have been kind of leading a paper around just identifying sample connections, like how do we actually anchor on a sample identifier to sort of start that whole process. And so working with NMDC and their sample submission process, working with the user facilities, the JGI and EMSL, to track the samples as they get turned into data. And then from there, trying to leverage mechanisms like the data transfer service we're building so people don't have to download those data and then re-upload them somewhere else. So this is a huge group effort from all the BER platforms that are working on this, not just me.
Thanks for watching this webinar. For more webinars and tutorials, subscribe to our YouTube channel. You can see announcements for upcoming webinars and recordings for previous webinars on our website at kbase.us/learn. Let us know in the comments what content you'd like to see in future webinars. Be sure to follow us on Twitter at DOE-KBase. And if you have questions or encounter an error when using KBase, please contact the help desk.